Thank you so much, David, for the introduction and the invitation. I would like to tell you a little bit about the work that we have been doing in our lab. I'm an engineer by training, by the way. I'm a chemical engineer who turned bioengineer. So I'm going to tell you about my perspective on solving an important problem in designing therapeutics. My lab works in the field of drug delivery, so that's pretty much what I'm going to talk about today. But an important aspect of drug delivery is dealing with the barriers that the body has posed. So I'm going to tell you about what those barriers are, why are they problematic, and what can we do to overcome them. But before we start talking about the future of therapeutics, I think it's worthwhile taking a look at the current landscape. What are some of the important drugs out there that have changed our lives? And depending on what your favorite drug is, you may have a different view on this, but maybe one way to look at this is look at the drugs which have been a huge commercial success. And these 5 drugs are the most blockbuster drugs to date that have been designed by the pharmaceutical and biotech industry. They cover a number of diseases, some common themes there. Cardiovascular comes up as a key area, arthritis, and to date they have made a lot of money to pharmaceutical and biotech companies, starting with Lipitor, the most blockbuster drug in history, has made about $150 billion, all the way to Plavix to $84 billion. And that is really a great story, because when I tell people, my friends and family, and tell them that I am working in the field of drug delivery, in their mind these are the examples that they see. They have impacted their life, they have improved their lives. So that's actually the positive side of what we do. But there is a dark side to what we do, and the dark side being that the cost of developing new drugs is currently very high. Now these numbers I have gotten from a reference, and numbers might be different depending on who you talk to, but roughly it takes about $3 billion and 12 years to make a new drug. And when your drug is successful, like the ones I listed here, that's wonderful. But these are only few examples. A lot of drugs don't make that much money. So when you look at the cost to benefit ratio of the pharmaceutical and biotech industry, that does actually raise a concern as to how can we sustain the kind of innovation that we need to move this field forward. This $3 billion is a lot of money, and I think it's very interesting to put that in perspective. So this thing here, this is Mars rover Curiosity, and it took off from planet Earth. It's landing on a foreign planet, moving around, taking pictures, sending data, and the cost of developing this thing is about $2.5 billion. So isn't it interesting that it's cheaper to send something to a place that most of us have never seen, only seen in pictures, and which is millions of miles away, but it takes as much money, if not more, to send something down our throat into our body to treat a disease. And why is that? I guess maybe a simple answer is that we understand the laws of physics much better than we understand laws of biology, but nevertheless to me that's a big problem. How can we understand body better, and how can we make sure that we can make the drugs in a more effective way and cheaper way? Now how do we do that? How can we bring the cost of drug development down? There are many aspects of that problem, and again depending on who you talk to, you might get a different answer. But to me the art and science of developing a new drug is really fighting this war between safety and efficacy. And what I mean by that is when you are doing your experiment in a petri dish on a bunch of cells or on a model substrate and trying to look for a molecule that can become a drug, there are many success stories. Now chemotherapy drugs, for an example, there are many molecules that can kill cancer cells in the test tube, but when you try to take it to animals and humans, that's where most things fall, because the barrier or the burden of safety is so high that we are not able to dose the patients at a dose that we need to make the drug effective. And whenever there is a tug of war between efficacy and safety, safety always wins, because the first rule of drug development is that you do not want to do any harm. So because of that, many drugs that we hope will reach the patients are not delivered properly, are not dosed properly, and the burden basically leads to the fact that many drugs are essentially suboptimal in the way they are delivered. Again, maybe one simplistic example is that if all you want to do is kill cancer, if you can deliver that drug selectively to the tumor site, many more drugs can be effectively used for that application. So, why is it that it is so difficult to control where the drug goes into the body? And that entire problem comes from biological barriers. The body is not designed to distribute the drugs the way we want. It is designed to control its own metabolism, and there are very complex set of principles that dictate that, and most of the time when you deliver drugs, you are fighting those and not using those. Let me give you a few examples. So, body's outermost barrier, skin, one of the most formidable barriers that the body has built, because the skin's job is to keep things out, and without that we would not survive. If we lose the ability of the skin to form the protection layer against the pathogens, against water loss, we would dehydrate in a very short period of time. So, thankfully, because of the skin, we are all alive. But now imagine trying to deliver drugs across the skin does not allow it. There are very few small molecules which can cross the skin if you put on it, but try putting a protein or a peptide, which is really where the therapeutic landscape is going to. Less than 1% of the topically applied drug will enter the skin if you are trying to deliver a protein. The problem continues if you try to deliver the drug by a pill. Wouldn't it be nice if you can deliver proteins orally? But when you deliver proteins orally, less than 0.1% of what you ingest will end up in the blood circulation, because the GI tract is not designed to transport proteins. It is designed to digest the proteins. So, whatever protein we consume, it is going to be digested into its building blocks, and even if some protein escapes from the stomach into the intestine, the mucus layer on the intestine and the epithelium of the intestine is going to make it very difficult for this protein to enter from the mouth into bloodstream. And again, for the most part that is good, because we do not want the proteins we eat to end up in the blood circulation, but if you are trying to deliver insulin orally for diabetic patients, you are not going to be able to do that if you do not design a good system. If you go inside the body, the barriers do not go away. For example, if you are trying to deliver a nanoparticle which can carry your drug, imagine injecting that in the blood circulation and you want it to go to a specific tissue, but the liver is going to take the biggest cut, because liver's job is to make sure that no foreign circulating particles exist in the body. So, most of the nanoparticles eventually end up in the liver and the spleen. So, if you are trying to target something like tumors, you are going to have very few particles left which can do that effectively. If you are trying to deliver the drug into the brain, which is again one of the most formidable barriers that the body has built. Brain is a highly privileged organ, and it does not really want things from the blood to freely enter into the brain, and it has built a very sophisticated barrier which not only prevent the entry of foreign things into the brain, but whatever enters the brain is pumped actively back into the blood. So, very advanced barrier built between the brain and the blood, which makes it very difficult to deliver drugs into the brain for the treatment of neurological disorders. So, when you look at these numbers, 1%, 0.1% delivery efficiencies, they really bother me as an engineer. When you look at other parts of engineering where people have developed new technologies like transportation, for example, if I were to tell you that you can take a taxi, but only 1% of the time you will reach your destination, we would never take that taxi. If you think about airplanes, that level of efficiency is not acceptable, but somehow in the field of drug development, this low efficiency which has become a building block, instead of trying to overcome it, we try to work around it, and that ultimately leads to high cost and long development times. So, our goal in the lab has been to understand how can we overcome these barriers, develop mathematical models to predict them, and develop new tools and technologies to overcome these barriers, so that we can deliver the drugs through the route that we want, to the location we want, and with those sort of technologies, I think the cost and time of drug development can come significantly down. Another key challenge that we are going to face in the field of drug delivery is that the therapeutic landscape is changing dramatically as we speak. So, if you look at how drugs have evolved over a period of time, if you go back in history, most drugs were small molecules, right? They were extracts from plants, natural molecules, and I wouldn't say it was easy to deliver them, but easier than the drug that we are facing today. The biggest challenge there was how to solubilize those molecules like paclitaxel. So, a lot of effort early on was focused on figuring out solubilization. Then came small peptides and proteins like insulin almost 100 years ago that increased the challenge of delivery. Then came antibodies, which are not only much larger in size compared to typical proteins like insulin, but they are also required in large doses. So, that adds another layer of complexity. Then came nucleic acid, where you not only had to deliver them into the body, but you basically had to deliver them across the cell membrane and sometimes into the nucleus. So, that adds another layer of complexity, and more recently cells have really come to the table as therapeutic agents. So, these are living entities. When you deliver them into the body, they can do their own things. So, as you go from small molecule to cell, your requirements change dramatically. So, our delivery technologies have to really take that into account and be as sophisticated as the drug itself. And because that landscape is changing constantly, it is needing constant innovation in terms of technologies, and that I think is a key challenge for people who are working in this field. So, let me give you a couple of examples as to what kind of drug delivery technologies we are working on and where they are in terms of their impact. One of the key areas in the lab has been trying to figure out how can we deliver proteins and peptides across the skin. So, things like insulin, can we develop a patch, a transdermal patch for insulin? There are some patches out there like nicotine, testosterone, estrogen. You can buy a patch for those, but they are all small molecules. If you try to deliver something large, it is not going to cross the skin. Why is that? Because the skin is a fantastic barrier. What you see here is a cross section of skin, and if you look at the topmost layer of the skin, this layer here, this is the main reason why your skin is a good barrier. That is the part that flakes off. It is only 15 microns in thickness, so extremely thin, but it is a fantastic barrier to transport of molecules into it. Once the drug crosses this topmost 15 micron, it can easily go through the rest of the skin and get absorbed into blood circulation, where the blood capillaries are located right in this region. If you look at the structure of that topmost layer, it is literally like a wall, where the bricks are made up of dead cells which are filled with proteins. In this case, it is mostly keratin, and the space in between those bricks is filled with lipids. And these are not ordinary lipids that you find in cell membranes. These are the lipids which have very long chain, and what that means is that they exist in a very solid gel like phase, and that makes them very difficult to cross. Even one lipid bilayer of this kind is enough to stop the entry of the drug, and in the case of skin, the drug has to cross about 50 to 100 of those before getting into blood circulation. So, that is the level of challenge we are dealing with for transdermal delivery. So, we have been figuring out how to overcome this, and the first tool that we developed makes use of ultrasound. So, this is the device that actually came out of the research. It is a device about this big. This particular piece sort of sits on a desk, and there is a hand piece. You put that hand piece on the skin wherever you want to make the skin permeable, and when you turn the device on, it sends sound waves at a high frequency, the ultrasound waves, and in about half a minute you make the skin permeable. About this much spot on the skin is made permeable, and that spot remains permeable for about 18 to 24 hours, and during that time you can put the patch. The drug diffuses across that open spot and enters the blood circulation. The way it works is that ultrasound causes cavitation. It forms pockets of gas. So, these gas bubbles form on the surface of the skin, and they expand, and at one point they collapse, and that collapse is very sudden. It sends a shock wave, and that shock wave can make the skin permeable. So, this device was approved by the FDA for delivering local anesthesia. It has been used to deliver a number of other molecules. It's been tested in clinical studies to deliver vaccines. Many other studies have been done with additional molecules which are difficult to deliver. About a couple of years ago, this device was used in another clinical study. In this case, it was a different challenge, but also dealing with the barrier. So here we were trying to use ultrasound for the treatment of acne. And the idea was the following, that when somebody has acne, the sebaceous gland, the sweat glands are overactive. They are producing a lot of sebum, and that blocks the channel, and that leads to the formation or the growth of bacteria and eventual inflammation. So we wanted to send these nanoparticles into the deep glands, and these are core shell nanoparticles. They have a silica core, and surrounding the core is a shell of gold, and when you hit them with laser, the gold shell absorbs the laser and becomes extremely hot, and the temperature of that nanoparticle can become hot enough to basically locally char the tissue. But it is extremely local. So, even a few microns away from that nanoparticle, you do not feel any heat. So, it is a very local, very focused way of inducing heat or imparting heat to the tissue. The only challenge is that how can you get those nanoparticles into the gland where it needs to work, and that is where ultrasound came to the rescue. We put nanoparticles on the skin, applied ultrasound, and particles were delivered deep into the gland, and then we zap them with laser, and you can essentially kill the local sweat gland, and that has beneficial effects. So, this was tested in a number of human volunteers and was published a couple of years ago, led to very interesting benefits in terms of the appearance and the progression of acne. But we were also interested in developing simpler to use formulations, and by formulations I mean a gel or an ointment that you can put easily on the skin that has its own benefits in terms of reaching to a large skin area, in terms of simplicity, in terms of cost of development. And we are wondering what would be a good sort of foundation to develop a sort of a technology like that, and we started thinking about ionic liquids in that context. So, ionic liquids are basically liquid salts, just like sodium chloride, except that in the case of sodium chloride you have a sodium and a chloride ion. They are very small, so they form a solid. If you take a large cation and large anion, they form a liquid. And we did a lot of studies to understand how do the properties of ions relate to their outcome, and out of that study came one particular ionic liquid, which is a combination of geranic acid and choline. And we like these two molecules for a number of reasons. So, geranic acid is a natural molecule. It is present in oranges and lemongrass, and it is currently used as a food flavorant. It has a very citrusy kind of aroma to it. So, it is used as a food enhancer or an aroma enhancer. And choline is present in a number of natural ingredients, predominantly eggs, but more importantly it is taken as a dietary supplement by a large number of individuals. So, we said that is interesting. If you take these two molecules with a history of use in humans, and by combining them, if you get a new ionic liquid that can do something very interesting, that would be a fantastic opportunity. And we did that, and out of that came the ionic liquid that we call CAGE. It is an acronym for choline and geranic acid, and it looks like this. CAGE is a viscous liquid, almost like a honey consistency liquid. In that bottle there, the liquid only has two molecules, choline and geranic acid, but the way they come together, they form this very interesting liquid which can solubilize drugs, which can enhance the transport of drugs into the skin. And one molecule that we are very interested in delivering is insulin, as I mentioned early on. So, we said let us see if we can deliver insulin with CAGE. So, we took CAGE, we added insulin to it, and in the beginning we did a simple transport experiment. We took pig skin and put insulin on the surface of the skin, and here is the control experiment where we put insulin in saline. Hardly anything goes through. That is to be expected because insulin has essentially no skin permeability, but when we deliver insulin from CAGE, we see deep penetration into the skin. here up to about almost a millimeter into the skin, we see insulin entering in significant quantities, which is very difficult to do. So, that was very exciting. Is it really biologically active? So, we did an experiment in a rat model where we put insulin in cage on the rat, and that data is shown here. So, if you look at the red graph, that is the glucose level of the rat where we have put insulin in cage on the surface, and you see a steady decrease in the glucose concentration, and the glucose concentration remains low for a long period of time. So, you can get that sort of sustained delivery of insulin across the skin into blood circulation. And in contrast, if you just use a saline solution, you hardly see any penetration. The blue line here is the positive control. So, if you inject it, that obviously works, you do see a drop in glucose level, but you see a quick drop and a quick recovery. So, there is a sort of a sudden change in the glucose level. In contrast, if you put a patch on the skin, you can potentially get sustained release over a long period of time. So, that is where a transdermal patch can make a significant impact. Not only it can deliver the drug into the body, but it can change the rate at which it goes into the body. It can also provide a new feature in terms of therapeutic benefits. Oral delivery, as you know, has been a sort of a holy grail, so to speak, where question is, can you deliver proteins and peptides using a simple pill? Now, pill has its own appeal in the sense that we are all used to taking pills, vitamins, headache medicines. So, if we can deliver many molecules that way, the thought is that that would be certainly welcome from the patient. So, here I am showing you some data where we made a pill that contains same cage ionic liquid, and in which we are suspending insulin, and we are delivering that to a rat in this case. And if you look at the graph on the right hand side just for the moment, what it basically shows you is that compared to a quick drop that you see in the case of injection, you can get a sustained drop in glucose levels when you deliver this orally. So, it was very interesting because there are very few technologies that I am aware of where the same kind of technology platform works against multiple platforms or multiple barriers. In the previous slide, I showed you that it enhances transport across the skin. Here it is enhancing transport across the GI tract. So, it has some very interesting features that we are currently exploring. In the context of oral delivery, cage does a few things. It is a good enzyme inhibitor. So, it inhibits the enzymes in the stomach. It is a good mucolytic agent. So, when the drug reaches the mucus layer, it can thin it out and enhance transport across the mucus layer, and it also is able to open the tight junctions in the epithelium, so can enhance the permeation. So, it can address all key barriers that are faced in terms of protein delivery. When you go inside the body, your barriers certainly do not go away. As I mentioned, they exist in different form. So, that is also something we are quite eager to explore, and the thought here is that you would take a drug, encapsulate that in a nanoparticle. It could be a polymeric nanoparticle, could be a hydrogel, could be a liposome, and you want that nanoparticle to go to a certain tissue in the body. Now, obviously, that does not happen as effectively because nanoparticles get cleared, and while we are looking at it, a question we started asking is, why is it that there is such a huge contrast between how the body deals with foreign entities like nanoparticles compared to its own cells? If you think about circulating cells, each one of them has a feature that we wish and hope that our synthetic nanoparticles can accomplish. For example, if you want your nanoparticle, if you want your drug to stay in circulation for a long period of time, look at the red blood cells. Each red blood cell stays in circulation for about 4 months, and after its time is up, it gets cleared. But there is no therapeutic, there is no small molecule, there is no large molecule, no nanoparticle which can come close to staying in circulation for 4 months. If you want your material, your therapeutic to bind selectively to blood vessel or somewhere in the body, look at what platelets do. So, platelets are inactive in their native state, but whenever there is damage to the blood vessel, they get activated, they bind selectively and amplify the signal to really induce the formation of the clot. So, that self-amplifying, highly targeted mechanism of binding is inherent to the platelets in a way that is very challenging for a nanoparticle to mimic. If you want your particles, your therapeutic to go deep into the tissue, look at what macrophages do. Now, macrophages stay in the circulation for a long time, their job is to do immune surveillance, but whenever they sense something wrong in the tissue, they escape the blood vessel and go deep into the tissue in a way that nanoparticles cannot. If you think about how drugs and nanoparticles navigate themselves in the tissues, they do so by diffusion, and diffusion is extremely slow, so it takes a long time for them to go deep into the tissue. So, many people who are developing therapies for cancer, they have realized that the drugs can access the surface of the tumor, but not deep inside because of the poor diffusion into the tissue, but macrophages have no such problem, they can go straight deep into the tumor. So, the question we have been asking is, what can we learn from these natural examples, and how can we adopt some of those principles into designing synthetic carriers which can deliver drugs in a better way. If you look at the contrast between natural cells like red blood cells and synthetic materials, what are the differences between these two systems which can drive such different response, and if we get better at synthesizing newer materials, and if we understand the biological systems in a better way, can we mend the two and begin to coexist in this intermediate range where we can use biomimetic strategies to overcome some of the key hurdles in drug delivery. Now, when you look at these two systems and you say what is different, and you say, wow, a lot of things are different, their surface is different, their composition is different, but as an engineer I really got intrigued by the differences in the physical properties. Their shapes are different, their flexibility is different. So, question we have been asking is, these physical parameters, do they really make a difference in terms of their behavior in the body? So, the first step was to make synthetic particles that can span different shapes, which is very difficult to do, because if you are making a material or making a particle, a sphere is energetically most favorable shape. So, going away from that always poses a challenge. So, one of my talented graduate students, Julie Champion, who is a faculty now at Georgia Tech, she figured out a way to make particles of different shapes, starting from discs to rods to UFO shaped particles if somebody wants them. And then she started asking, does it really make a difference? And one of the first experiments she did was she fed them to the macrophages. Long story short, it turned out that it made a huge difference whether or not particles went inside the cells in a way that depends on the shape. So, if you have a macrophage and if you have a rod like particle that approaches the cell with the pointy end, it got internalized very quickly, but if the same particle comes from the side, it was not able to get internalized. So, from the cell's point of view, shape made a huge difference in terms of the outcome of this interaction. And that led us to all kinds of interesting questions as to how do other aspects of particle interaction are impacted by the shape. We found out that if you take an antigen like a vaccine and put that on the particle, the way the immune cell perceives that vaccine is very different depending on whether you have a sphere or a rod like particle. And that may have some origins as to how pathogens are processed by the immune system, where pathogens that come in very unique shapes could have some potential advantage in terms of their inherent immunogenicity. The way the body generates an allergic response to nanoparticles also depends on shape. So, those of you who are active in the field of nanomedicine may be aware of this, that allergic reaction to nanoparticles is a huge concern for some of the nanoparticles, and a couple of years ago we showed that simply taking a sphere and turning that into a rod like particle, you can essentially mitigate most of the allergic response in the animal model. Same thing happens with respect to particle softness. So, here we made particles of the same material, but we changed their softness. We made a soft version of it which is almost like a gel cell membrane like elasticity, and we made a hard version. And when we inject these two particles in the blood, we found out that the softer particles circulate for a much longer time. They escape the clearance by the immune system much more significantly, and that basically gave us an idea as to how important these elastic properties are, which in hindsight makes sense because most materials or most entities in the body are soft, made from either proteins or lipids, and compared to that most synthetic materials are very hard. So, inherently they stand out as foreign entities. So, softness is a key parameter in sort of our mind. So, when you combine these two parameters, the shape and the elasticity that is inherent to biological systems, we started wondering if we put them together, can we make some interesting materials. So, our first attempt at that was to make synthetic red blood cells, and by that I mean particles that physically look like red blood cells. So, the picture on the left, those are our particles SRBC. So, they are synthetic red blood cells made from proteins and polyelectrolytes, and on the left hand side you see natural mouse red blood cells. So, if you compare the morphology of these two, you can begin to imagine the structural similarity that we are quite excited about. Just like natural red blood cells, these soft particles can squeeze themselves through small capillaries. They can carry oxygen if you load hemoglobin into them. So, you can begin to build sort of complex architectures and functionalities into these particles. Going one step beyond, we wondered whether we can mimic platelets to make synthetic platelets, and that is an important medical problem because there is currently no synthetic material that you can inject in the blood as a replacement for natural platelets. If somebody is bleeding profusely, there is no treatment other than natural platelet that you can use, but the problem with natural platelets is that you cannot store them. So, that is always a challenge. So, we made these particles. These are completely synthetic particles made using albumin. They have a similar size and shape like natural platelets, and we put targeting ligands on them to make them bind to the blood vessel when it is damaged, and we did a simple test to see if they stop bleeding. So, compared to a saline control, these synthetic platelets were able to reduce the bleeding type dramatically, and currently we are building on to that to see if we can move them to the next stage of development. While we are trying to mimic the principles of natural cells, we realize we are not quite there yet. So, it is more of a lifelong query than a quick fix, and we wondered while we are working on it, can we somehow make use of the circulatory cells to bring on some of these functionalities into synthetic particles. So, for example, if we cannot design a nanoparticle that can stay in circulation for a long time, can we just make them hitch a ride on the red blood cell? So, we designed nanoparticles which attach themselves to red blood cells. So, you can see a red blood cell with number of particles attached to it, and the underlying question was actually quite fascinating. Now, imagine that complex of red blood cell and nanoparticles arriving in the liver, and the macrophage is looking at it, and it sees a foreign signal coming from the particle and a self signal coming from the red blood cell. Who is going to win, and what tips that balance? So, it led us to a very interesting set of questions which I will be happy to discuss with you at a later time, but the first key question we wanted to ask is, does it stay in circulation? So, compared to free particles which get cleared from the blood before you know it, this is an experiment in a rat. So, these free particles almost most of them got cleared even before we could measure. If you simply put them on the red blood cell, they stay in circulation for a much longer, and if you put polyethylene glycol on the surface of this exposed particle, you can keep them in circulation for even longer. You can keep them in circulation for days. So, that really opens up new functionalities from these particles. So, what happens to them eventually? The particles do come off over a period of time, and what happens is that when these complexes of red blood cells and nanoparticles go through small capillaries, the red blood cell gets stretched and this particle gets attached to the endothelium, the surface of the blood vessel. So, it is a great way of transferring a particle from the red blood cell to the surface of the blood vessel, and the red blood cell itself is basically let go, it continues to circulate. So, if you look at the circulation of red blood cell itself, it is not impacted. Turns out that this strategy has been used by pathogens. So, what you see here is an electron micrograph of Haemobartonella. It is a bacterium that is found in cattle, and it has found a way to attach itself to red blood cell, and by doing so it can avoid clearance from the circulation. So, some really smart pathogens have figured it out way before we did. The strategy works for a variety of particles. So, here we have used hydrogel nanoparticles, liposomes, polymer particles, protein particles. They all work by the same principle because it is a very physically driven mechanism, not sort of chemistry specific. And these particles tend to go to the lungs. They do so because lung has largest number of capillaries per unit mass. So, when these complexes go to the lung capillaries, the particles are dislodged from the red blood cell by the endothelium. So, it is a great way of targeting nanoparticles to the lungs. If you do not want lungs, if you want them to go somewhere else, you can do that as well. So, here we inject these red blood cell nanoparticle complexes in the artery, and then they go to the brain. So, here we are getting about 10% of the injected dose to go into the brain, and the significance of that is that that number is among the highest fraction of nanoparticles delivered to the brain compared to what people have seen. Even the best performing antibody that people are discovering for the purpose of entering the brain is not able to come even close. So, we are very excited about the potential of targeting nanoparticles to different organs based on this principle. Once you like hitching a ride, why stop at red blood cells? Start hitching a ride on all kinds of cells, and we like to ride on macrophages and monocytes. And we like them because they know how to get deep into the tissues. So, here we figured out a way to attach a particle to macrophage. The difficult thing about macrophages is that unlike red blood cells, which are happy to take your particle, macrophages want to eat it because that is their job. So, we figured out that if you make a disc shaped particle and that is flexible, it can attach to the macrophage but not get internalized. If you give them spheres, they will just gobble it up. And this little particle can go for a ride wherever the macrophage goes. The macrophage really holds on to it, so it does not go anywhere, and it is thin enough so that it does not hinder the macrophage. So, it is a great way to get into different tissues in the body. So, what we are doing with it right now, our main target is to use this for the treatment of cancers, with the idea that if you take a large tumor, any typical large tumor, it has macrophages in massive quantities. And that happens because macrophages is a key part of body's innate immune system. So, when tumor develops, these macrophages go in there, and monocytes as well, and their initial approach is to attack the tumor, but the tumor overrides that signal and they become essentially part of the tumor because they are very plastic in nature. So, we are delivering cytokines from these backpacks so that we constantly reminding the macrophage to take on an anti-tumor phenotype so as to launch an anti-tumor response. So, currently we are in the middle of these studies, and I hope to have more to tell about that in the coming months to a year. Where we see this field overall going is that we as sort of material scientists have made a lot of progress in making different kinds of materials, liposomes, nanoparticles, and the interface between the material world and the cellular world coming from red blood cells, macrophages and T cells, they existed in two different parts of the world. But as we get better and better in terms of designing complex nanoparticles and either using them as an inspiration to make these particles or simply hitch a ride on them for the time being, we can begin to blend the gap between these two, and that I think really opens up new opportunities, especially as the world of therapeutics is beginning to think of cells as a key part in the drug landscape. So, let me end my talk here and maybe save some time for questions. This is the slide I showed you early on in terms of the biological barrier that we are facing. With the technology that we had developed in the lab, we are making significant progress in terms of overcoming these barriers. So, for skin, we have gone from 1% to about 20% in terms of delivery of insulin and nucleic acids. For intestine, we have gone from very low number for oral uptake to high double digit transport of proteins. In terms of delivering particles within the body, less than 1% going to the tumor and other target tissues, we can go up to a high number in terms of targeting, same thing about the brain. So, that is a significant improvement in the delivery efficiency across the barriers. But the bigger impact I think of these numbers is that the current therapeutic world is very focused in terms of how certain molecules can be delivered. For example, when somebody is developing a protein drug, you immediately think about only injections, automatically discounting the possibility to use transdermal or oral because of these high barriers. And once you start crossing these barriers, that really opens up in an impacting way. And once you take away that stereotype in terms of which route can be used for which molecule, that really enables not only delivery of these molecules, but also impact discovery of new drugs. And that is where I see the future and a huge impact of delivery technologies on the therapeutic landscape. So, let me end here. I want to end my talk by thanking you for all coming here on a Wednesday evening. I want to thank all the students who have done this wonderful work that I presented. The top picture is a drawing by one of my students. So, one fine day I go to the lab and I see this drawing on the fume hood, and it shows how our lab works. So, everybody in the lab has a job. So, somebody is responsible for purchasing supplies, one person is responsible for injecting animals, and this is me, and my job is to stay in my office and say sounds great, which is exactly what I do. So, I am deeply thankful to all their hard work, and thank you again. Thank you, Dave, for the invitation, and if there is time, I will be happy to answer questions.